Okay. Good evening everyone. Everyone please confirm me. Good evening everyone. Please confirm me. Am I audible to everyone? Everyone just confirm me. Am I audible to everyone? Okay. Divya, please confirm. Am I audible? Rohini, please confirm. Am I audible to everyone? Thank you, thank you. All right. Now let's continue without further delay. All right, let's continue first. Later we will discuss if anyone having any kind of a doubts. Okay, I will discuss 20 minutes continuously. After 20 minutes, if anyone find any kind of a doubt, you can ask me immediately. All right. So friends, if everyone quickly recall, in the last class we have completed all the lipids that are present on the plasma membrane. We have understood very clearly all the type of proteins present on the plasma membrane, especially variety of proteins that are present on the plasma membrane. If you consider the types of proteins on the plasma membrane, we understand integral membrane protein, peripheral proteins, as well as transmembrane proteins, and we understand their functions also. Later we have started studying about the membrane carbohydrates. Every one of you very quickly answer, all the online people, what is the major function of the carbohydrates on the cell membrane? Everyone please confirm what is the major functions of the carbohydrates on the plasma membrane. I believe everyone remember the major function of the carbohydrates on the plasma membrane is what, friends? Cell-cell communication or the cell-cell recognition. Please do not get confused over there. Compared to the lipids, everyone listen friends very carefully. Compared to the lipids, compared to the proteins, the total percentage of the carbohydrates on the plasma membrane is extremely lower. Hardly only 5 to 8% is present. If everyone recall my words, I told to all of you some of the carbohydrates which are present on the plasma membrane, some will be attaching to the lipid, some of the carbohydrates attaches to the protein. Whatever the carbohydrates attached to the lipids, we call it as the glycolipids, and whatever the carbohydrates that are attached to the protein, we call it as the glycoprotein. Now what is the point that I want you to remember for the exam? When compared to the glycolipids, glycoproteins present most abundantly in the plasma membrane, and I believe every student remember this is the last slide that we have discussed in the last class. What we understood, the sugars or the carbohydrates attaches to the plasma membrane via two type of linkages. I believe everyone remember. Please continue all of you. One is called as O-linked glycosylation, friends, and another one is called as what? N-linked glycosylation. I believe everyone remember this particular diagram without fail. These are all the glycoproteins, correct na? Can you see friends, this is the protein and this is also another protein. So protein, in its active site there is a presence of certain amino acids. You know that very well. Now listen, in that particular active site of the protein, there are two amino acids. Especially, I believe everyone remember friends. One is called as serine amino acid and another one is called as threonine amino acid. When the sugars are added via serine and threonine, that kind of addition of the sugars to the protein, we call it as O-linked glycosylation. There is no need to get confused over there. Similarly friends, whenever sugars are added to the protein via nitrogen as their major linkage, we call it as the N-linked glycosylation. Here, what kind of amino acid majorly involved here? The amino acid involved is the asparagine, because compulsory you may get the questions from the either locations, most importantly the type of amino acids involved over there. Never ever confuse friends. Now all of you pay attention. When you consider the N-linked glycosylation, all the students listen. Where is the site? Listen everyone. where is the site that N-linked glycosylation is generally taking place? All the students remember, N-linked glycosylation is exclusively taking place, because this is the question directly we can expect in the exam. That is ER lumen, or the inner space of the ER, the N-linked glycosylation is taking place. When you understand O-linked glycosylation, O-linked glycosylation exclusively taking place in the Golgi complex. There is no need to get confused with that particular point. I want you to understand that very correctly without having any confusions. Now everyone listen my statements correctly, okay? Because the kind of questions are like this. All of the following functions are performed by the carbohydrates on the cell membrane except. So it is very important you have to remember exact functions performed by the carbohydrates on the membrane. So now listen, most of the carbohydrates present on the plasma membrane, everyone focus friends, they will make up of the glycocalyx. Whenever you heard the name of a glycocalyx, always remember it is made up of sugars plus proteins. It is the combination of sugars also, proteins also. What is the major function of this particular glycocalyx? All the students remember, it keeps the cells moist and slippery. You know, right friends, bacterial cell, especially bacterial cell, in order to maintain the moisture conditions, in order to maintain the moisture environment, always they will use this particular glycocalyx. Not only for that, so suppose example, one bacterial cell wants to be connected to the another bacterial cell, sometimes they will use lectins as their major receptors. Suppose example, just understand a condition. This is a one cell, okay? Now this is a bacteria. If the bacteria want to attach to this particular cell, they will attach to the cell via lectin as their major receptor. So in simple words, remember friends, glycocalyx which are present on the bacterial membrane, majorly it is required for cell-cell attachment and cell-cell recognition. No need to get confused over there. Now listen, the glycocalyx is a carbohydrate enriched coating, covers the outside of many eukaryotic cells and prokaryotic cell, especially in the bacteria. Now, whatever the function performed by the glycocalyx in the prokaryotic organism and the eukaryotic organism, slightly different. But if you consider the eukaryotic organism, the glycocalyx majorly required for cell-cell recognition or the cell-cell attachment. But if you consider the bacterial cell, everyone remember, it is not only required for the attachment or recognition purpose, especially in the bacteria, the glycocalyx can act as a protective coat. What does it mean? Every student please understand my statements very correctly. Friends, let's consider this is a bacterial cell. Every one of you understand. This is a bacterial cell. Let's consider friends, this particular bacterial cell is surrounded by this glycocalyx. Now what happens whenever this particular glycocalyx is surrounding the bacterial cell, especially our immune cells? You take an example of macrophages, you take an example of dendritic cells, which generally attack on the pathogens. They cannot recognize. Once again understand, our immune cells such as macrophages and dendritic cell, they cannot recognize the cell or they cannot recognize the bacterial cell which is surrounded by the glycocalyx. So what I can say here, the possession of a glycocalyx on bacteria is associated with the ability of the bacteria to establish an infection. What does it mean? Whenever our macrophages or dendritic cells are not able to recognize this particular glycocalyx surrounded bacteria, bacteria can escape from our immune defense mechanism. Whenever it escapes from immune defense mechanism, it can easily establish an infection. Don't get confused. Read 2 to 3 times, then only it becomes easy. Whatever the glycocalyx that is present in the bacteria, it generally exhibit in 2 forms. Number 1, everyone remember, One form of glycocalyx is a condensed form. We also call it as a tightly arranged form. Then another type, we call it as the loosely attached form. One is called loosely attached form, another one is called as the condensed form. You already read multiple times even in the 11th standard also. Whatever the glycocalyx that is attached very tightly, that is called condensed form. The best example what we can take, a capsule. What is that? Capsule. So friends, capsule nothing but what? It is a type of the glycocalyx only. You know, right? Capsule will help the bacteria to tolerate in the adverse environmental condition or in the unfavorable condition. That is the ultimate function of the glycocalyx, which is arranged in a condensed form or tightly packed, which is called as capsule. In some bacteria, this particular glycocalyx, instead of arranging very tightly, they arrange extremely loose. So the loosening form of the the loosening form of the glycocalyx present on the bacterial cell wall, we called slime layer, or we also call it as what layer? S layer. What do we call it, friends? We can call it S layer or slime layer if the glycocalyx is loosely attached. Friends, in case if the glycocalyx is very tightly attached, or if it is in the condensed form, we call it as the capsule. There is no need to get confused over there. Now all the students, please try to understand this unique statement. All of you pay attention towards this. There are two prominent functions of the glycocalyx. Everyone focus. It enables the bacteria to become harder for the phagocytic uptake. What does it mean? What is the meaning of this line? Inside our body, macrophages, dendritic cells, what they will do, whenever any kind of a pathogen particle enters in our body, they will hold it, they will capture, they will do the process of phagocytosis, and they will try to destroy that particular pathogen. But whatever the bacteria that is surrounded by the glycocalyx, it can easily escape. It becomes almost invisible to the immune cell. It becomes more invisible to the immune system of a host cell. It can escape from the immune system, and most importantly, it can establish or it can cause the infection at a higher speed. Don't get confused. That is about glycocalyx. Now all the students understand my statements very correctly. Consider, friends, this is one bacteria, this is second bacteria, this is third, this is fourth, this is fifth, this is sixth, seventh, and so on. Correct? Friends, whenever there is a group of bacteria are present, every student understand my point very correctly. Whenever a group of bacteria are present, every bacteria contain glycocalyx. There is no doubt about it. Every single bacteria contain glycocalyx. Now all these bacteria are present in a culture. Now when all the glycocalyx of a various bacteria come close together, they can form a very rigid, compact structure. We generally call it as biofilm. What is that? Biofilm. So how can you define the word biofilm? Many bacteria, the cell, what we can say, the glycocalyx of the many bacteria come close together, they can form a rigid, compact structure. We call it as a biofilm. What is the speciality of this particular biofilm? Biofilm bacteria can become very hard to kill because of the presence of a glycocalyx. Glycocalyx itself biofilm, but many glycocalyx of many bacteria come close together, they can form a rigid structure, that is called as the biofilm. Now whatever the bacteria that are having the biofilm, they can escape from the immune system, they can cause persistent infections. What is the meaning of a persistent infections? The infections occur very oftenly and very continuously also. Immune system also fail to recognize it. Even though immune system recognize it, it cannot destroy that type of bacteria which contain the biofilm. So the bacteria which contain biofilms, they generally cause the persistent infections. There is no need to get confused over there. So friends, what we discussed so far? So far we discussed what are the type of proteins present on the membrane, what type of lipids present on the membrane, what type of carbohydrates present on the membrane. Okay, anyway 90% of you people studied type of protein, studied type of lipid, studied type of carbohydrates. Now we are going to study a very important topic, friends. Very importantly, you have to remember for the exam, that is fluidity of the membrane. I want every student has to remember this. Whatever the plasma membrane is there, it never be stagnant, which means that it never stick to one place. So plasma membrane always in a fluidizer manner, it is moving like this. It is always in the movement, it is always moving. Now the point is here, how does the plasma membrane maintain that particular fluidity or the movement? Of course, you all people know very well, the fluidity can be managed by the saturated fatty acid. There are two type of fatty acid, you all people read, saturated fatty acid and unsaturated fatty acid. You people read in the 11th standard also. Now every student listen. I told to all of you multiple times, you generally know that compared to the saturated fatty acid, undoubtedly friends, unsaturated fatty acids will increase the fluidity. What is the reason why unsaturated fatty acids increase the fluidity compared to the saturated? Every student please look into the picture. If you observe the unsaturated fatty acid, look into this. It contain a bent. It contain what? It contain a bent or a kink-like structure. Can you see this picture very clearly? All of you observe the picture. If you observe unsaturated fatty acids having a double bond, because of the double bond, it can form a kink or a bent-like structure. Because of the kink or bent-like structure, can you see friends, there is a lot of space got accumulated. Because of that space, fluidity will be increased. There is no need to get confused over there. But what about the saturated fatty acids? Every student remember, you all know very well, saturated fatty acid only contain a single bond. So can you see the picture? Because of the presence of the single bond, all the saturated fatty acids are arranged in a tightening form, very compact structure. You can see there is no gap was observed within the saturated fatty acid. As a result, compared to the saturated fatty acid, unsaturated fatty acids only will increase more fluidity on the membrane. Anyway, that is easy friends. Now every student listen my question very carefully and try to understand what exactly I am trying to explain to all of you. Can you see here? You know very well plasma membrane is made up of what friends? Plasma membrane is made up of lipids. Whatever the lipids that are present on the plasma membrane, they will be keep moving. They will be keep moving from one place to the another place. Most of the lipids that are present on the plasma membrane, they generally shows two type of a movements. Every student pay attention and look into the picture. Correct, very good. So most of the lipids present on the plasma membrane will show two type of a movements. Everyone focus on this. Number one movement, we called it as friends lateral movement. What is the meaning of a lateral movement friends? Everyone pay attention. Whatever the lipid that is present on the one layer, can you see the picture? It is moving onto the same layer but at a different position. Again, every student please try to understand my point. Whatever the lipids that are present on the one layer, the lipid is moving on to the same layer but at a different position. An example friends, this is the lipid 1, this is the lipid 2. What is happening now? Lipid 1 is moving to the place of the lipid 2 and lipid 2 is moving to the place of a lipid 1. This kind of a movement where the lipids are moving on the same layer, we call it as the lateral movement. This is the movement occur very oftenly. I can say to all of you friends, the lateral movement occur nearly 10 power 7 times per second. that much oftenly this kind of a movement will occur. Now friends, next very important movement, we call it as the flip-flop movement. This is also you people can expect the question in the exam. What kind of a movement it is? Flip-flop movement. What is the meaning of a flip-flop movement? This is one lipid bilayer, this is another lipid bilayer. Whatever the lipid present on the top layer, it will come to the below layer. Whatever the lipid present in the below layer, it will come to the top layer. You can see this picture, friends. This is one lipid and this is the another lipid. This is present in the top layer, this is present in the bottom layer. Now, the lipid present on the top layer, it will come to the bottom position. The lipid which is present in the bottom position, it will come to the top layer. So this kind of a movement, we call it as the flip-flop movement. Now friends, please don't think that it will also occur very often, which is absolutely wrong statement. Flip-flop movement occur very rarely. I can say to all of you, it happen only once per month. It happen how, friends? It happen only once per month only. There is no need to get confused over there. I'll quickly summarize. Plasma membrane maintain a structure, maintain a property called as fluidity. The fluidity of the membrane can be maintained by two type of fatty acids: saturated fatty acid, unsaturated fatty acid. Saturated fatty acid, due to its compact structure, due to the presence of single bond, they will always contribute to the less fluidity. When it comes to the unsaturated fatty acid, because of the double bond or because of the kink, it will increase the fluidity. In order to maintain the fluidity, lipids has to move on the membrane. How the lipids are moving on the membrane? There are two type of movements are there. One is called as lateral movement. What we discussed, friends, about the lateral movement? Lateral movement means lipid is moving on the same layer. But friends, when we come to this particular flip-flop movement, what is happening? Lipid present on the topper layer, it is coming to the below layer. Whereas lateral movement happen very oftenly, whereas flip-flop movement occur very rarely. I can say that flip-flop movement is happening only once per month. That's all. Now, no need to get confused. Now, here comes a question, very wonderful question. I believe every student in the online will give the answer for this question. Now, do you really think lipids can move on their own without anyone help? My question is, friends, do you really think lipids can move on their own without anyone help? Of course, yes. Lipids can move from one place to the another place on the membrane without anyone help also. But the point is that it always takes a lot of time. So that is the reason lipids always uses certain type of carriers in order to move on the membrane. Once again, everyone remember, in order to move on the membrane, in order to save the time, lipids always uses certain type of carriers. How many type of carriers are present on the plasma membrane? There are total 3 major type of carriers which are involved in the transport of a lipid. One is called as flippases. Second one is called as floppases. And third one is called as scramblases. Third one is what, friends? Scramblases. Flippases, floppases, scramblases. What kind of a question you people can expect in the exam? You can get a question like this. Which of the following lipid can be transported by scramblases? Or among the flippases, floppases and scramblases, which are calcium dependent, which are calcium independent? Or among the flippases, floppases, scramblases, which are ATP dependent, which are ATP independent? This kind of questions very oftenly they will ask in the exam. Anyway, if you read the PPT, that is more than enough. You can tell lot of information. What I want you to remember is which carrier is transporting which lipid. That's all I want you to remember. You people for DL level, you no need to read extreme depth over there. So listen to me carefully. Flippases. I'll use a word. Flippases either ATP dependent or ATP independent. Friends, what is the meaning of this word? Sometimes they are ATP dependent, sometimes they are ATP independent. Whenever they are, listen friends, very carefully understand my statements. Whenever they are ATP independent, we cannot define which lipid it is transporting specifically. What does it mean? Whenever it is in the ATP independent manner, it always follow non-selective fashion. Friends, what is the meaning of non-selective fashion? It is not choosing the lipid very specifically. Whichever the lipid is available, it will transport that. But if the flippases are ATP dependent, friends, whatever the lipid it wants to be transport, it will choose very specifically. Let me explain it with the help of a example. All the students listen. Flippases, sometimes they will be ATP dependent or sometimes they will be ATP independent. Sometimes they will be selective. What is the meaning of selective? I already told you now. Very specifically they choose the lipid to be transported. Whether it want to transport phospholipid, choline, sphingolipid, cholesterol, what it has to be transported, it will choose very selectively. Whenever they are using ATP. Whenever it is not using ATP, it will transport the lipid randomly. Let us understand more deeply. Every student focus on this picture without having a confusion. Focus on the picture. Friends, this is the lipid. Can you see that? This is the lipid. Now this lipid is showing flip-flop movement. What does it mean? This lipid from the top layer, it is entering into the below layer. You can see the picture. Finally, can you see this picture, friends? Finally, lipid entered into the below layer. From the top layer, lipid molecule entered into the below layer. Here it is not at all using any kind of a carrier. Here it is not at all using any type of a transporter. And friends, how much time it has taken? It has taken half day. It has taken half day for one single lipid molecule to transport from top layer to the below layer. But all the students focus. But same lipid molecule, whenever it uses the flippase, can you see the picture? Whenever it uses the flippase, it is transporting to its targeted area in half a second. It is reaching to its targeted area within half second. So what you have to understand, lipids can move on their own also, but most of the time, in order to save the time and for the rapid movement, they always uses the flippases. Flippases are involved in the flip-flop movement also. Flippases are involved in the lateral movement also. But compared to the lateral movement, friends, flippases are majorly involved in the flip-flop movement. No need to confuse it, easy to understand. Now just now I told to all of you, friends, flippases are sometimes ATP dependent and sometimes they are ATP independent. Correct na? Whenever flippases are ATP dependent, this is where you can expect the question. Whenever they are ATP dependent, they will choose the lipid very specifically. They will only transport phosphatidylserine and phosphatidyl ethanolamine. Phosphatidylserine and phosphatidyl ethanolamine. Only these two type of lipids can be transported by the flippases in the ATP dependent manner. There is no need to get confused over there. That is about first type of a transporter, which is called as flippase. Now second kind of a transporter, floppages. Once again listen my statement. Just now I told to all of you, whenever we read the flippages, sometimes it is ATP dependent and sometimes it is ATP independent. Now you listen. Whenever I use the word floppages, every student remember, floppages are strictly ATP dependent. What does it mean? If the floppase want to transport any kind of a lipid, mandatorily they are going to use the ATP molecule. There is no need to get confused over there. Listen, floppases use the ATP to transport the substrate. So floppages are actually transporting what kind of a lipids? Phosphatidylcholine and sphingolipids. From which direction to which direction? Against the concentration gradient. Every student please look into the picture, friends. This is the diagram of a Flippase. It is moving the lipid from which direction to which direction? From outer direction to inner direction. Everyone please try to understand. This is the direction. You can see the picture. Friends, when you look into the floppages, what you have to observe? Floppages are involved in the movement against. What do you mean by that? Inner to outer. Correct na? Suppose example, friends, you are standing in the ground floor. Everyone listen. You are standing in the ground floor. You want to throw a stone, a small stone to the 5th floor. You cannot throw very easily. You have to use abundant amount of energy in order to throw a stone from ground floor to the 5th floor, because you are throwing against the concentration gradient. Similarly, friends, whenever the lipids are moving against the concentration gradient, undoubtedly they will use ATP as their major source of energy. There is no need to get confused over there. Now listen, understand my statements very correctly. Floppases having a other name. Very good, very good. Floppages are having a other name called as ABC transporter. Why we have given the name ABC transporter? ABC represents ATP binding cassette proteins. What proteins, friends? It is ATP binding cassette proteins. That is the major function of the floppages, also called as ABC. Why we are calling ABC? Can you see the picture? It provides a binding site for the ATP molecule. That's why we can call it ABC transporters. Now the last and final type of the lipid transporter, which is called as the scramblases. I will compare the 3 transporters once again. Scramblages are strictly ATP independent. Strictly ATP independent. Again, remember, scramblages are also involved in the movement of amino phospholipids only. Especially, we gonna discuss in the other chapters about this, especially in the biochemistry lipids concept. The scramblase is exclusively involved in the transport of a very important lipid called as phosphatidylserine, which is exclusively important for the blood clotting purpose also. Now, I will summarize, friends. You no need to remember whatever I have told you so far. Instead of all the things, you just remember this particular slide enough. When you are doing the revision or understanding, you just understand this slide very correctly. Can you see this picture now? If you observe the flippase, it is involved in the transport of ions or molecule from outer to the inner. What kind of a molecules or ions it is transporting, friends? It is transporting phosphatidylserine and phosphatidylethanolamine. First point. And second point, you remember sometimes they are ATP dependent, sometimes they are ATP independent. That's what I want you to remember. When you heard the name of a floppages, what do you have to remember? Floppages are also said to be the ABC transporters. They strictly uses the ATP molecule, and they are involved in the movement which to which direction? From inner direction to the outer direction. What kind of a lipid it is selectively transport? It will transport phosphatidylcholine and sphingolipids. And when you look into the scramblase, mostly it won't use the ATP. It is ATP independent. Mostly it is involved in the transport of cholesterol molecule in both directions. In both directions, it is involved in the movement of cholesterol and another lipid named as phosphatidylserine. Now, again, the most important question I want all the students has to remember: among the flippases, floppases, and scramblases, which type of a transporter is calcium dependent? Because this is also a very important point you can expect as a direct question. The only transporter that is strictly calcium dependent, that is only scramblases, right? Do not get confused. So friends, this is about structure, composition of the plasma membrane. We understand about lipids, we understand about proteins, we understand about carbohydrates, we understand about the fluidity of the membrane also. Now, before reading the functions of the plasma membrane, what is the major function of the plasma membrane? Every one of you know that transport. So it will show passive diffusion, passive transport, active transport, primary active transport, secondary active transport. Before understanding the transport mechanism, we have to read some more cell organelles. Then when we read all those cell organelles, understanding the transport mechanisms are also very easy. So first we will read some more organelles, then we will come back to the transport mechanism. So this is about structure of the plasma membrane. Now let's focus on the next organelle, that is the nucleus. Okay, every student please confirm, is it clear so far, the plasma membrane? Everyone quickly confirm. Every one of you please quickly confirm, is it clear everyone, up to the structure and composition of the plasma membrane? Whenever you are reading also, you read the PPT with you. Always read the PPT with you, and I will provide the PDF. You just read that PDF. It is 100% sufficient. Then go and solve the question. You no need to do anything extra. It is very easy to understand also. Alright. Is it clear everyone till here? Everyone please confirm me, is it clear everyone? Okay. If you are not responding, I never come to know that whether you are getting the concept or not. We should be always engaging, you are learning something. Okay, not a problem in that. Okay. Now, the second major organelle that we have to focus is the nucleus. Again, whenever we are reading the particular organelle nucleus, what exactly we have to read? Again, we have to focus on structure. Very important. We have to focus on functions. Most important, where we people can expect the question in the exam, is the transport. These three are the major mechanisms or major processes we have to read whenever we are reading the cell organelle nucleus. Shubham, we are having, okay, this is not the set class, okay, we are having, okay. Now, listen, now once we consider the nucleus, if I ask you a simple question to all of you, what is the major function of the nucleus? Can any one of you please answer my question? What is the major function of the nucleus? Everyone. You know, we use it to say that brain is the major organ which will control and coordinate every activity in the body. Correct na? Similarly, friends, when you consider a cell, within the cell, nucleus is said to be the brain of the cell. Correct na? When you consider a cell, within the cell, nucleus is said to be the brain of the cell. Why we say like that? Because nucleus is the one which will control, coordinate every single activity happening within the cell. How we can say that nucleus is only controlling and coordinating every activity that is happening inside the cell? What is the reason? The reason is very simple, friends. The nucleus contains the genetic information, which is our DNA. So DNA is present within the nucleus. We know very well what is the ultimate function of the DNA? Synthesis of a protein. Protein is the one which will control and coordinate every activity of the cell. So that protein is synthesized by the DNA, and that DNA is present within the nucleus of the cell. That's why nucleus is said to be the most important organelle which control and coordinate every activity of the cell. Now, what are the major functions? I hope if you quickly recall molecular biology also, we read multiple times. Ultimate function of the nucleus, it stores the cell's hereditary material or DNA. I don't know how many of you remembered when we are reading the molecular biology, we read very clearly. DNA replication, transcription, correct na? RNA processing, I believe every student remember very correctly. RNA editing, all these processes are strictly happening where? All these processes are strictly happening within the nucleus only. Correct na? That's why in the eukaryotic organism we say nucleus is the area where the replication, transcription, post-transcriptional processes, RNA editing, everything will happen over there only. Okay, we will read in the upcoming slide also very elaboratively. And I believe you all people read in the 11th standard and 12th standard only elaboratively. Within the nucleus only the three type of RNAs are getting synthesized: mRNA, tRNA as well as rRNA. All the type of RNAs are getting synthesized within the nucleus only. If you quickly recall about the rRNA, you know very well rRNA is getting synthesized in the nucleolus. And when you consider the tRNA, when you consider the mRNA, these two are getting synthesized where, friends? I believe you all people remember, within the nucleoplasm. Correct na? Nucleoplasm is the area where mRNA, tRNA is getting synthesized. rRNA is getting synthesized in the nucleolus. But what is the common point? Whether it is rRNA, mRNA, tRNA, all the type of RNAs are getting synthesized within the nucleus only. There is no need to get confused over there. Now friends, how does the nucleus look like? What are the various active components of the nucleus? Everyone listen. Every student please look into this beautiful picture and everyone just try to understand what exactly I am trying to explain to all of you. We read in the 10th standard also, nucleus is what, friends? A nucleus is a bimembrane organelle. Can you see that? We generally call it as the outer membrane and we call it as the inner membrane. And you know very well the space between the outer membrane and intermembrane, we generally call it as intermembrane space. And you also know very well when it comes to the nucleus, friends, we don't use the word outer membrane, we don't use the word inner membrane more oftenly. Instead of using the word outer membrane and inner membrane, both membranes together, we call it as nuclear envelope. Everyone understand my point very correctly. Instead of using the word outer membrane and inner membrane, we use a simple word called as a nuclear envelope. So nucleus contain nuclear envelope. Every student pay attention towards the picture very correctly. This nuclear envelope at a specific locations. All the students look into the picture. This nuclear envelope at a specific location, they can form a pore-like structure. You can see that. They can form a pore-like structure, which we generally call it as what pores? Nuclear pores. Everyone understand my statement. Nuclear membrane or the nuclear envelope, it is completely impermeable. What, friends? It is completely impermeable. It never allow the movement of any kind of a ion, the movement of any kind of a molecule, because the membrane is impermeable. Then how does the transport generally happens within the membrane? The transport generally happens within the membrane only via nuclear pores. There is no need to get confused over there. And can you see this, friends? The concentrated area where the chromatin is present. Everyone listen. The concentrated area where the chromatin is present, we generally call it as what? Nucleolus. What you people has to observe, in this particular nucleolus only, what will happen, friends? The rRNA synthesis will take place within the nucleolus only, which means the protein synthesis is generally happening within the nucleolus. Do not get confused over there. Now, apart from the nucleolus, I want every one of you has to understand a rigid structure, not rigid, a gel-like structure which is present within the nucleus. We call it as a nuclear matrix. Now, if you ask me a question, the difference between the nuclear matrix and nucleoplasm, all the students remember, nuclear matrix contain proteins. Along with the protein, it also contains some of the inorganic substances also. Whereas if you observe the nucleoplasm, friends, in the nucleoplasm you can observe only proteins. You cannot observe any kind of inorganic substances within the nucleoplasm. Only inorganic substances are occurred in the nuclear matrix. Now listen, all the students pay attention towards this very important diagram. I want all the students pay attention towards this diagram. Everyone, focus on this picture. This is called as outer membrane. This is called as inner membrane. Correct na? So outer membrane and inner membrane together only, friends, we call it as the envelope. Correct na? Every student observe the picture correctly, friends. The inner membrane of the nucleus, can you see this picture, friends? The inner membrane of the nucleus can be connected via mesh network like structure. What, friends? The inner membrane of the nucleus can be connected via mesh thread like structure. I believe everyone remember. What do we call this mesh thread like structure? We call it as the nuclear lamina. Friends, what is the major function of this particular nuclear lamina? Everyone remember, if the nuclear envelope wants to become stable, what, friends? If the nuclear envelope wants to become stable, compulsory they require the nuclear lamina. Stability of the envelope can be mediated by this particular nuclear lamina. And what does this particular nuclear lamina is made up of? The nuclear lamina is made up of exclusively a protein named as laminin protein. Don't confuse and don't forget, friends. Now, can you see this picture, friends? At a particular location, nuclear envelope, I mean to say, outer membrane and inner membrane is not fused at a specific location. The area where both membranes are not fused, they are going to form a nuclear pore-like structure through which complete transport happens within the nucleus. There is no point of getting confused over there, and it is very easy to understand also. Now, everyone look into the picture, friends. What you people observe here, the outer membrane of the nucleus, I am only talking about outer, not the inner. The outer membrane of the nucleus can be connected to the rough endoplasmic reticulum. So we all people know very well, the rough endoplasmic reticulum is studded with lot of ribosomes. That's the reason it is involved in the synthesis of a protein. So don't confuse that. Only outer membrane of the nucleus is connected with the endoplasmic reticulum, not the inner membrane of the nucleus. Don't confuse over there. So once again, I will quickly summarize. The outer membrane of the nucleus and inner membrane of the nucleus, we call it as the envelope. The envelope becomes stable with the help of a network of thin thread structures, which we call it as a nuclear lamina. And what we discussed, friends, the nuclear lamina is made up of a protein called as a laminin protein. Now listen. And only the outer membrane of the nucleus can be connected to the rough endoplasmic reticulum, which is studded with the ribosome. So friends, everyone listen, very important statement. So inner membrane, outer membrane, this is called nuclear lamina, correct na? Now understand my statements. If nuclear lamina collapse, undoubtedly nuclear envelope also collapsed. Which means, friends, if the entire nucleus wants to be stable, compulsory we have to maintain this particular lamina. What we discussed, friends, lamina is made up of what protein? Laminin protein. This laminin protein can be regulated by two type of mechanisms. One is called as phosphorylation mechanism and another one is called, friends, dephosphorylation mechanism. Don't forget, phosphorylation and dephosphorylation will regulate the functioning of this laminin protein. Now every student understand my statements very correctly. You imagine a condition, every one of you imagine a condition. There is a mutation in the laminin gene. Whenever there is a mutation in the laminin gene, there were two type of diseases will occur. One is called as muscular dystrophy, number one disease. And friends, number two disease is said to be the Hutchinson-Gilford progeria syndrome. What are the major differences between these two? Muscular dystrophy means, friends, muscular dystrophy is a condition of loss of muscle mass. What, friends? Loss of muscle mass. So, muscles will become completely brittle. If you just apply a little bit force on the muscles can easily tear. Right? So, that is what loss of muscle mass, which is also called as muscular dystrophy. Now, the most important disease I want all of you to remember is second one, Hutchinson-Gilford progeria syndrome. Friends, what is this particular Hutchinson-Gilford progeria syndrome? Every student try to understand my statements without having a confusion, friends. What is the specialty of Hutchinson-Gilford progeria syndrome? Listen, everyone. We call it as early aging. What do we call it? Early aging, which means 10 years old boy look like a 50 years old person. You might observe in Bollywood movie also far. So what happens over there? Because of the early aging, even though the age is 10 years, they will appear like a 50 years old. That syndrome only we can call Hutchinson-Gilford progeria syndrome. So these two diseases are generally caused by the mutation in the laminin genes. There is no need to get confused over there. Okay. Is it clear all of you so far? Is it clear all of you so far? Anyone having any kind of a doubt, you people can ask. Clear all of you? Okay, okay. Not a problem. Okay. Okay. So from tomorrow onwards, friends, 5:15 to 6:30 a.m. Okay. The class will happen in the YouTube until the cell biology chapter get completed. That's all. And tomorrow evening 4:30 to 6 p.m. we will be starting the biochemistry for both botany and zoology people. I will give the separated timetable as well. Okay, it is strictly to the app only. Okay, only one chapter of the cell biology I will teach through the YouTube, only one chapter. Okay, so whomever wants to be enrolled, please enroll within 2 to 3 days. Okay, even though the batch is 40 to 50, it's enough. Okay, I'll help every one of you until you crack the exam. We will discuss very briefly and very elaboratively in the classes. Okay, so tomorrow the timings are fixed, 5:15 to 6:30 a.m. in the YouTube only, the chapter of cell biology until it get completed. After that I don't do any kind of a classes in the YouTube. Then 4:30 to 6 p.m. every single day, Monday to Saturday or Monday to Friday, depending on the convenience, we will be starting biochemistry. We will keep the target of June 8th or June 9th. For botany also as well as zoology also, we will complete the entire syllabus by June 8th and 9th. Okay, whomever wants to enroll very faster. Once after 1 or 2 days I will go very vigorously and we have to cover the syllabus very fast in the specified time interval. Okay, if you have any kind of a doubt, you can ask me via app or you can ask me via WhatsApp. Okay, thank you all, thank you.